Hey everyone, welcome to Professor Long's Lectures on Anatomy and Physiology. As you guys know, we're in the coronavirus shutdown, so I'm doing these videos online. These videos are intended for use by students who are enrolled in my Human Anatomy and Physiology course, so it's targeted to those students. If anyone else out there in YouTube land finds these helpful, by all means use them. But know that even within our own institution, each one of us may emphasize different areas of the topic, go in a little bit more detail here, a little less there. So these videos are, again, intended for my students, the way that I teach the course, for what I want them to know. If you find them helpful, use them, but know that they may not be thorough for your own particular instructor. Now, what we're going to be doing today is the first in a series of videos on the endocrine system, or our hormone system. I have some definitions on the board. I'm going to go over those with you. And then what we're going to do is we're going to do sort of an overview of how the overall endocrine system works. Some of the definitions on the page, if you're following along in my class, you should have the note set. There is a preview of what we're going to cover on page 20. We're going to start on page 21 with these definitions. And then I'm going to step back and do a broad overview of the endocrine system, and then we'll go into some detail. Then the following videos will go into very specific hormones and their modes of action, how they're released and everything. So for the definitions, at least for now, you guys need to know these definitions, and there's some others you could know, but these are the ones I want to focus on. So y'all should know the definition of homeostasis. Homeo means same, like homo, and stasis means to stay. So it really means to stay the same. And the technical definition that is often used is the maintenance of a constant and optimal internal environment. Essentially, if it ain't broke, don't fix it. So if your body and your cells are functioning just right, we don't want to change the temperature, the oxygen or glucose or carbon dioxide or any of the other parameters, the ion concentrations or anything. If it ain't broke, don't fix it. Now, the reason we talk about homeostasis is it's the central tenet of anatomy and physiology, and particularly of healthcare. When patients fall out of homeostasis, they are in pathology, a disease state. It's not easy for the cell to survive, and if it continues, the cell could die, then the organ dies, the tissue suffers, and so on and so forth. So one of the things we have is positive and negative feedback. Now, those definitions aren't on the board here, and they're not in the note set at this point, because you should have already covered them at some point. But ultimately, you need to know that in positive feedback is a response to a stimulus that increases that stimulus. One good example is labor contractions. When the first one hits, over time, the pain, the contractions increase in frequency, in intensity, and in duration. So once the first one hits, then more come. That's a type of positive feedback. Negative feedback is a response to a stimulus that decreases or removes the stimulus. So if we have sort of some kind of negative stimulus, something that's making things worse, for example, if it gets too hot, your body has mechanisms to cool you back down so that your cells can perform their chemistry at optimal temperatures. And so that would be a type of negative feedback. Most feedback loops are negative feedback, especially in the endocrine system. So you should know those two definitions, although I don't have them written down. Now, in exocrine secretions, exo means exit or to leave outside, and crine means to secrete. So it literally tells you these are secretions onto the external surfaces of the body. And in these secretions, if I were to draw one, I'm going to do it here in black ink. Usually you have some kind of surface of the body. You have a long tube leading to that surface called a duct, and then you have some kind of ball of cells or a bunch of structures lining this little tube, and when they make secretions, those secretions come out onto the external surfaces. Okay, these secretions exit the body somehow. Things like mucus running out of your nose, saliva. If you fall asleep in class, you wake up and there's drool hanging onto your books. That's an exocrine secretion. All of your digestive secretions. We tend to think that the digestive tract, the inside of it, is inside of our body, but it's really not. If you swallowed a pen, it would come out the other end looking like a pen. It doesn't ever enter your body. It passes through this tube. I like to tell my students that you're one funny-shaped donut where the whole twists and turns, but it runs all the way through you. Okay, so sweat onto the external surfaces. A lot of our reproductive secretions are exiting the body. Okay, So all of those would be exocrine secretions. The glands for exocrine secretions almost always have a duct. Well, they do always have a duct that goes to some external surface. Now, the opposite of that is endocrine secretions. Endocrine secretions, the definition we tend to use is these are secretions released directly into the bloodstream or into surrounding interstitial fluids or extracellular fluids. A good example would be our hormones. In this instance, I'm going to draw on the same space. Now, I could have a surface somewhere in the body here. I have a group of cells, a little endocrine organ. They're going to release a substance. Usually what happens is that substance is going to enter the bloodstream and go to some very distant group of cells. And as it travels through the bloodstream and pops out, the cells that have receptors for it will get the signal, and then they will respond to that. Notice the hormone never makes it to the outside of the body. Usually endocrine glands are ductless glands. They don't have ducts that go to the external surfaces. And eventually the hormone is broken down. It never exits the body. So we call these endo, which means into, crine, secretion. So endocrine secretions are released directly into our bloodstream. These are onto the external surfaces. Got it? Now, a hormone is defined as any substance released by a group of cells that has what we call a tropic effect or tropic effect on a distant group of cells. This group of cells releases this compound, and by the way, this is going to be my abbreviation for hormone. I tend to abbreviate it as an H with an R. When I was a student, we didn't have handouts of PowerPoints and stuff. You just had to write what the instructor was saying and writing on the board, or was already on the PowerPoint, or usually an overhead projector. That's how old I am. And so I developed my own shorthand so I could keep up with it in my notes, and I got tired of writing the word hormone out, so that's my personal abbreviation for hormone. It's not some accepted abbreviation. Okay? So when a hormone is released by one group of cells, it will bind to and alter the physiology of a different group of cells. The word tropic or tropic means to change or changing. Like a tropical rainforest or a tropical island is a place where the weather is just constantly changing from morning to midday to afternoon to night. It's constantly changing. So tropic means changing. So we can say that this has a tropic effect on a distant group of cells, or another way to say it would be any substance released by a group of cells that alters the physiology of a distant group of cells. It makes them change what they're doing to restore homeostasis. Now, I did skip the definition for stimulus. You should know the definition of a stimulus. I mean, we did this in part one a bunch of times, which is any detectable change in the environment. Okay? So a stimulus is any detectable change in the environment. So in most instances, these cells will not release the hormone unless they are stimulated by something that they detect, some change in the environment around them. For example, we have hormones that are going to lower the calcium concentration in our body. So there's a group of cells somewhere in the body, an endocrine organ, that is monitoring calcium concentrations. If calcium becomes too high, these cells will release a hormone that will bind to other cells that will take up the calcium or cause it to be released out of the body so that we lower our calcium concentration. Okay, we'll talk about the details of that stuff and which hormones involved later. But ultimately, some stimulus, some change causes an endocrine organ to release a hormone. That hormone will go over a long distance, usually through our bloodstream, bind to some cells called target cells, and cause the target cells to alter their physiology, which leads us to another definition. Target cell. It's any cell that has receptors for a specific hormone. So if we were talking about a hormone called calcitonin, I could say the target cells for calcitonin are found here and here in the body, in the bones and in the kidneys. And you would know that the cells and some of the bone cells and some of the kidney cells have receptors for the hormone calcitonin. So we're going to be using these definitions and talking about these words a whole lot. Now, the next three definitions I didn't write because we're going to talk about them in detail in just a moment. Membrane receptors, intracellular receptors, and intranuclear receptors. Ultimately, when we get to the target cells can have a receptor in one of three locations. Okay, it can have a receptor in the membrane. We call those membrane receptors. It can have a receptor in the cytoplasm, which is called an intracellular receptor, and occasionally there's hormones that have receptors inside the nucleus of the cell, which are called intranuclear receptors. There's a few that even have receptors inside the mitochondria. So we'll talk about the location of the receptors for certain hormones. I'm going to get into detail in that in just a moment, okay? So now, I'm going to erase this. Those are the major definitions I want you to know. They're all on page 21 of the note set. You can pause the screen and write this down. You could also look these up in the textbook, which I highly recommend that you do. Let me erase this, and let me do a broad overview of how the endocrine system in general works. This is very generalized, and then we're going to go into individual hormones in the next video or two. I'm going to talk about the overall function of the endocrine system, and I'm going to talk a little bit about intracellular, intranuclear, and membrane receptors. So now, when it comes to how our endocrine system functions, essentially somewhere in the body will be a stimulus is detected. There's some kind of change happening. Somewhere in the body there's a group of cells, usually bundled together, and these would be considered to be what we call an endocrine organ. Okay? When these cells detect that change, they can release a substance called a hormone. Again, that's my abbreviation for hormone. It's an H and an R stuck together. Now here's what happens. Here's what's unique about the endocrine system, and we're going to compare it to the nervous system in just a second. This compound will enter our bloodstream, and it's going to go anywhere where blood vessels go. I'm going to draw 5 different branches off of this blood vessel, okay, just to illustrate something to you. When the hormone enters our bloodstream, it will go, some of it can go down each one of these blood vessels, and eventually it will, I say pop out, it will be released from the bloodstream at small specialized blood vessels called capillaries. Now we haven't done blood vessels yet, but capillaries are very tiny vessels. They're the only vessels that allow any substances to enter or exit the bloodstream. So when this hormone exits at some capillary, it's going to enter into the interstitial fluid in these other areas of the body. This particular hormone is going to go to 5 different areas. Now when I look at the cells at the end of the pathway, these cells can have receptors either in the membrane, inside the cytoplasm, or inside the nucleus. I'm going to draw these as membrane receptors for this simple setup. Now the receptors in the cell membrane can have different shapes, and they can have many different receptors. If the hormone's 3-dimensional shape and its electrochemical properties are such that it won't bind to this cell is not a target. Now, if this cell has a receptor that has the appropriate shape, and it can have some other receptors for other chemicals, but if that hormone can bind to this cell, this would be called a target cell. And eventually the target cell is going to give some kind of response. It's going to alter physiology, so we call that a target cell. Let's say this cell does not have the same receptors. It's missing the ones that fit this hormone. This cell would not be a target cell. It couldn't respond. If this cell and this group of cells over here all have receptors and the hormone can bind to them, these would all 3 be target cells. So I can have one hormone released. It can go to multiple areas of the body and alter the physiology of multiple groups of target cells in multiple locations. Now, another type of cellular communication in the human body is synaptic transmission or synaptic communication. In synaptic communication, okay, because this is going to be called endocrine communication, right? We're talking about endocrine system. So in endocrine communication, a hormone, in response to a stimulus, a hormone is released into the bloodstream and can target multiple cells at multiple locations in various parts of the body at great distances, and it will bind to those target cells, the cells that have the appropriate receptor, and all of these cells would give the proper response, okay? Now, in synaptic transmission or synaptic communication, one of the differences is I have a neuron release neurotransmitter, remember my abbreviation from part 1 for neurotransmitter is an N and a T, directly onto the next cell, or a very small group of cells in one location. If I have neurons over here, and neurons over here, and another neuron way up here, this neurotransmitter is not a very hardy or durable substance. Plus, we have enzymes in this synaptic cleft that will digest the hormone immediately. The neurotransmitter immediately. So neurotransmitters can't drift off. The only cells that they can communicate to are the ones that are directly within reach of the synaptic cleft, or right in that synaptic cleft. So it's almost directly onto the next cell that we're releasing neurotransmitter. It goes over a very short distance. Now, we can compare and contrast endocrine communication, or endocrine responses, and responses to neurotransmitter and synaptic transmission. And one of the things we know is this. So, in endocrine responses, one of the things we know is the response to the stimulus is slow. It could take minutes, it could take hours, it could sometimes take days. So we have a very slow response, where in synaptic communication, the response is rapid. It's instantaneous. And it's within the order of what we call milliseconds. So this happens, synaptic transmission can happen. When I stimulate this cell, I can get a response here within 1 to 2, sometimes 3 or 4 milliseconds, thousandths of a second, almost instantaneous. Where here it can take several minutes, up to hours, days, and weeks for us to get a response, okay? Now, another thing we know is that the duration of the response for hormones and endocrine is very long. They have a very long duration. As a matter of fact, when these cells get the hormone bound to them, when the target cells receive the signal, they can respond sometimes and release compounds that can have effects that last for days, weeks, months, years, a lifetime. Think about our sex hormones. When we start releasing them in high concentrations, when we hit puberty, our bodies change, and those changes last the rest of our lifetime. Okay? Once a female develops breasts, they don't go away. They're there for the rest of her life. Our body hair patterns, facial hair, they stick around for a long time. Where the duration here of the response is very short, usually on the order of a few minutes. If you almost have a car wreck and you release the neurotransmitter adrenaline, boom, your heart rate goes up, you have a very rapid response. If you don't wreck and you're safe, within a few minutes, you're back to your day. Your heart rate goes back to normal. So this duration can be on the order of minutes, a little longer, but this can last way longer. The other difference is here you're responding over a short distance. Here you're responding over great distances. There's a number of other things that we could discuss when it comes to comparing and contrasting synaptic and endocrine communication, but I want you to know this. We communicate over great distances to multiple sites, often multiple target cells in different locations. The response is rather slow. When we get a signal, it could take days, weeks, months for the response to occur, and then when the response does occur, it's very long term, which is very different than our nervous system, okay? So finally, what I want to talk about in this video is this, okay? I'm going to erase all this stuff. I need this board. I need some space to write and draw. So bear with me while I erase. Pardon my back. But this is how the endocrine system generally works. Okay. When we come to the target cells, so I'm going to talk specifically about target cells right now. At the target, our target cells have a cell membrane, they have their nucleus, and as you know, within the nucleus is where we store all the DNA. And you should know we have 23 pairs of chromosomes, 46 pieces of DNA, 23 times 2. Now, there's a series of bases on here that if I turn this gene on, there's a series of bases on a chromosome called a gene. A gene is the instructions on how to assemble a protein, and proteins kind of perform most of the functions within our cells. They're the functional molecules. If I need a specific function to occur inside the cell, I have to have the protein that performs that function. So I have to go turn that gene on in order to make that protein. Now, some proteins are being released daily in our cells because the cell has to do these certain functions every day. But every now and then there's a function that the cell does not need to do every day. It would be wasting time and effort and energy. But if the cell gets a specific signal that says, we need to do this now, sometimes that cell has to manufacture or synthesize that protein through transcription and translation. In transcription, we rewrite a gene of DNA into messenger RNA. In translation, we send that out to the ribosome, and the ribosome assembles the protein. So essentially, a gene on here is going to give us some kind of protein, in most instances, that's going to alter the function of this cell. Now, how do our cells know when to turn a protein on or turn a protein off? Because they get some kind of signal. Now, if this is my target cell, this target cell can have proteins that it's made through transcription and translation, protein synthesis, and that might be done on this particular gene here on this chromosome, where I'm constantly producing this protein, and this protein can be a membrane receptor. And sometimes, over time, since these are physical structures, just like a doorknob or something wears out and you have to replace it, these proteins are synthesized and put in the membrane. If they wear out, we can take it back in and replace it. So there's turnover of these proteins. All right. Now, I have all these proteins out in the cell membrane. And since they're embedded into the cell membrane, these are membrane receptors. And the way we describe a membrane, that's my abbreviation for the word receptor. I don't want to write it out every time. Membrane receptors are described as integral membrane proteins to which a hormone will bind on the surface of the cell. So the hormone, when it reaches here, can bind to a lot of these receptors. Now, when it comes to membrane receptors, there's a way that we signal the gene inside the nucleus. We can turn a gene on, or we could turn a gene off if we want to stop performing a function. So whether I'm going to turn this gene on or off depends on the hormone and the response and in the specific cell. But nonetheless, usually what's going to happen is somewhere closely associated to the membrane is going to be a thing called a G protein. And we refer to the G protein as a second messenger. The first messenger was the hormone. Now, something I've explained in my part 1 class, and you'll see me talk about several times, is that when proteins are manufactured, I'm going to give you a little bit of background, and then I'm going to move on to this stuff. When we synthesize a protein is nothing more than a whole bunch of amino acids on a long chain. Each individual amino acid gets stuck to another one in a long string of amino acids, almost like beads on a necklace. Now, when we study the structures of amino acids, there's only 20 amino acids that all human proteins are made out of. The difference between one protein and another is the number of amino acids and the order and the arrangement of the amino acids. Now, amino acids, when you study them in biochemistry, have these little side groups. And let's say these two amino acids have side groups that if I put them close to each other will automatically form or instantaneously form a chemical bond. And since these proteins are floating around in fluid like pasta in a pot when you're boiling it, eventually if I bring those two amino acids close to each other, it will fold up and create a chemical bond there, which might bring two other amino acids close to each other, and then the protein will fold some more, which will cause almost like when the best example I can give you is sort of like those windshield visors that are round, and once you twist them, they collapse automatically. Well, that's what's going to happen, is eventually the protein will fold up into a three-dimensional shape, and there might be a little loop of proteins that might bind to another protein somewhere else in the body. So eventually I can have one protein or one substance bind to another protein. And when they do, these amino acids in here are going to break one bond and bind to this, which might cause other parts of the protein to unfold. So anytime I stick a protein to another molecule, another part of that protein might be released or change its shape. So proteins are changing their shape or conformation as they're binding and unbinding to each other. The reason that's important is because when I start to break a bond and unfold a protein, as it changes shape, it may now open up some amino acids that can alter or communicate or bind to or affect another molecule. So watch. That's probably not the greatest explanation, but let's just say I have a protein that's shaped like this, and I have another substance that can bind to it. When they bind, if I break these chemical bonds, then another part of the protein might change its shape as it's bound here, okay? When I unbind them, it may fold back up. So proteins changing shape, got it? Well, what happens when this hormone binds here is as this protein changes its shape, it activates or interacts with the G protein. That G protein is an enzyme, usually it has enzymatic activity, which might take another molecule, let's say substance X, and convert it to something else, and then that's going to take another molecule and convert it to something else. So it sets off a series of chemical reactions. Once the hormone binds to the membrane receptor, it alters its shape and triggers a G protein or second messenger. That G protein will set off what's called a signal transduction cascade. Signal transduction is the conversion of a stimulus into a physiological response. And we call it a cascade, or sometimes we just say it's an enzyme cascade. Now, a cascade is a series of steps, like a cascade would be with dominoes falling in order. So this little cascade of enzyme reactions, eventually one of those enzymes will enter the nucleus, bind to our gene, and either turn it on or turn it off, altering the cell's physiology. So here's what you really need to know. Now, the protein shape was just to help you understand how it occurs. But what you need to know is this. When a target cell has a membrane receptor, that membrane receptor is an integral membrane protein. It's embedded in the membrane of the cell, the external membrane, and that's where the hormone will bind. When the hormone binds, it alters the shape of the receptor, which triggers a second messenger called a G protein, almost always, and then that will set off a signal transduction cascade or a series of enzymes that eventually will turn a gene on or turn a gene off, altering the cell's physiology. I hope that makes sense. Now I'm going to go over an intracellular receptor. Let me erase some of this, okay? Matter of fact, I'm going to erase almost the whole thing, and I'll redraw it, okay? And I think the more you see this, the better it sticks to your brain. So here's my target cell. Only now I have a receptor somewhere in the cytoplasm of the target cell. And the cell can have different receptors. It can even have membrane receptors for other compounds. Now in this situation, the hormone will pass straight through the cell membrane, enter the cell, and bind to the receptor inside the cytoplasm. So in an intracellular, when we have a target cell with an intracellular receptor, or what they call a cytoplasmic receptor, the hormone passes through the cell membrane and binds to a receptor in the cytoplasm of the target cell. Okay? The membrane, the hormone passes directly through the cell membrane, will bind to a receptor inside the cytoplasm of the target cell. Now this can set off a signal transduction cascade, and eventually some enzyme will enter the nucleus and then turn a gene on or turn a gene off. This part is very similar. The only difference is this enzyme cascade, and sometimes the hormone receptor complex can enter the nucleus directly, But it does not involve a G protein called a second messenger. Never does, okay? Only membrane receptors do. Now, one of the things that's unique about this is this. If you know what the cell membrane is mostly made of, which is lipids, well, then the hormone is usually a lipid or cholesterol derivative, meaning it is derived from or synthesized from lipids or cholesterol. I mean, after all, if the cell membrane is lipids, how do you get into a cell membrane? You got to look like a lipid. You got to pass right through. Lipids can mix in with other lipids and pass directly through. If the hormone were a protein, proteins have a very difficult time passing through the lipid bilayer. Here's the example I like to use. If I wanted to sneak into a nun convention, what would I need to do? I'd have to put on a habit and dress like a nun so that I could walk up, oh, hi, good to see you, sister. Oh, no, good to see you. And then I could sneak in and go deliver a message to somebody. When it comes to membrane receptors, I'm going to draw this on the same picture, but this would not happen this way. On a membrane receptor, the hormone can't enter the cell. It binds to a membrane receptor, which tells a second messenger, which then eventually goes and delivers the message. This would be like me trying to get into a nun convention and someone saying, you're not allowed in. You're not a nun. You can't come in. So I might say, well, can you hand this to Sister So-and-so in room 28B? She's in this hall down there. And someone else can take it. They would be the second messenger. And they would deliver it to someone else and then to someone else. And eventually the message gets where it needs to be. Do you see the difference? I hope so. Now, the only difference for an intranuclear receptor, and I'm going to draw this on the same picture. In an intranuclear receptor, the receptor is inside the nucleus of the target cell. And that marker's not working. But the receptor would be sitting inside the nucleoplasm. And in this case, the hormone would derive all the way through the cell membrane, all the way through the nuclear membrane, and then eventually alter protein synthesis. All three hormones pathways, how they enter the cell or how they affect the target cell, are going to have the same result. They're going to turn a gene on or turn a gene off. But how they get there, how they get the message across, is dependent upon the location of the receptor. One last thing I want to point out to you. By the way, all intracellular and intranuclear receptors have to be derived from lipids or cholesterol. That's how they dissolve all the way through. Okay? If the hormone has a membrane receptor, that hormone, all hormones that have membrane receptors are derived from either amino acids or are small proteins themselves. So hormones that bind to membrane receptors and trigger second messengers are usually called amino acid hormones or peptide hormones. And that means they're made out of a single peptide, a single amino acid that's been altered, or they're a small chain of amino acids. They're a small protein or a small peptide. Okay. So three things. The location of the hormone. If it's in the membrane, it triggers a second messenger, will eventually turn a gene on or off through a series of enzymes.